Hello and welcome. This is our first video in our video series for lectures in Chapter 4, Genetic Inheritance. I am Miss Delia, and if you're ready, let's get started. So in Chapter 4, Genetic Inheritance, we have three main subtopics, which is these three, 4.1, 4.2, and 4.3, and we're going to focus on 4.1 today, which is Mendelian genetics, monohybrids, and dihybrids. And these are the learning outcomes for this subtopic. Now, it may look like a lot, but as we go through it, you'll realize that it's only terminologies. Okay, kita belajar beberapa kata kunci, and we'll talk about Mendel and his pea plants. And then we'll talk about monohybrids and dihybrids. Once you learn how to draw the genetic diagram, you'll see that 4.1 is actually quite simple. Okay, dia agak ringkas, cuma kamu perlu belajar dan tahu macam mana lukis genetic diagram. Okay, so if you are ready, let us get started on the terms. The first term that we learn about is gene. Gene means a basic hereditary unit consisting of a sequence of DNA that occupies a specific location on a chromosome. Ini dia punya definisi formal, tapi secara ringkasnya, apa itu gene? Dia adalah satu segmen DNA yang membawa maklumat. Okay, it carries some genetic information. If people ask you how to define gene, better if you can give this definition. Tapi kalau kamu mau ingat dalam otak, sekadar faham saja, gene adalah sebahagian daripada DNA kita. Dan dia ada lokasi yang spesifik pada DNA. Okay, the next term is allele. What is allele? Alleles are alternative forms of the same genes that will occupy the same position on a pair of homologous chromosomes. So have a look at this diagram here. Okay, this is two different chromosomes, but they are homologs to each other. Jadi dia orang memang satu pasang. Okay, on this first blue chromosome, you see that it has the gene for flower color. Dia ada gene untuk warna bunga, tapi allele pada kromosom biru ini adalah allele untuk purple flower. In the second chromosome, this red chromosome over here, the same location is the gene that codes for flower color also, but it has the different version, the alternative form, which is allele for white flower. So allele adalah gene untuk ciri-ciri yang sama, tapi versi berbeza. Okay, alternative form of same gene. And we also need to talk about locus. What does locus mean? It is the specific position of a gene or an allele on a chromosome. Secara ringkasnya, locus adalah location, lokasi di mana gene atau allele itu akan duduk pada kromosom. So as you can see here, the locus for this flower color gene, whether the allele is purple or allele is white, is this area, this position on the chromosome. Okay, let us move on. We'll talk about genotype and phenotype. We'll start with genotype. It is the genetic constituent of an organism. Ataupun kita boleh katakan, bila kita cakap genotype, kita tengok individu tu ada gene apa, ada allele apa. And phenotype is what you say is the observable characteristic of an organism. Apa yang kita nampak dari luar, apa yang kita boleh lihat, itu adalah phenotype. Now, I'm going to use an example here. In my example, I would like to talk about the genotype and phenotype of your chin shape. Kita akan tengok bentuk dagu kamu, ya. Okay, so I would like to use the gene for chin, and the alleles are gonna be big C or small c. You can have a genotype of big C, big C, small c, or small c, small c. And the phenotype is cleft chin. Okay, kalau kamu tengok dagu kamu, kalau dia macam ada garis yang memisahkan kiri dengan kanan, that means you have cleft chin. And if you have uncleft chin, you look at your chin, tiada macam garis tengah-tengah, dia tidak pisahkan kiri dengan kanan, itu adalah uncleft. I think mine is a little bit more of a cleft chin. Okay? So if you see here, your genotype can be either big C, small c, small c, small c. Your phenotype is whether you have cleft or uncleft chin. In this case, these two genotypes will produce this phenotype. This genotype will produce this phenotype. And we're actually going to go into that in the next segment. Okay, in this segment, let's talk about a few more terms, which is homozygous and heterozygous. What is meant by homozygous? Homozygous means they have identical alleles at a locus on a pair of homologous chromosome. Homo means same. So kalau depan belakang adalah sama, dia adalah homozygous. Kita tengok di sini, P besar dengan P besar itu adalah homozygous. P kecil dengan P kecil itu adalah homozygous juga. Heterozygous means that it has different alleles at a locus in a pair of homologous chromosome. Dua allele yang berbeza. Jadi kita tengok sebelah kanan ni, P besar, P kecil, that is a heterozygous genotype. Quite simple, quite straightforward. All right, next term is dominant allele. Now notice that I've been saying big P, small p. Big C, small c. Saya cakap besar dan kecil. Tapi actually, term yang lebih sesuai untuk digunakan adalah dominant ataupun recessive. Bila kita cakap dia big P, small p, itu lebih kepada macam kita cakap secara tidak formal. Okay, so formally it is known as a dominant allele. A dominant allele is an allele that is expressed when present in either homozygous or heterozygous condition. Sama ada dia dengan satu jenis dengan dia, for example, big P, ataupun dia wujud dengan versi yang berbeza dengan dia. Contoh dia heterozygous, big P, small p. Dominant allele will always show their phenotype. So as you see here, homozygous, dominant, big P, produce purple flower. Heterozygous means it has one dominant allele. It still produces purple flower. Opposite of dominant allele is recessive allele. Allele that is expressed when in homozygous condition but being hidden in heterozygous condition. Recessive allele dia mudah mengalah. Jadi bila dia dengan satu jenis dengan dia yang tidak suka bergaduh, homozygous recessive, baru dia akan tunjukkan phenotype dia. For example, in this one, small p will produce white flowers. But when it is with a dominant allele, recessive alleles will not show their phenotype. Bila dia dengan dominant punya allele, dia akan mengaku kalah dan dia tidak akan tunjukkan phenotype dia. Okay, let us move on to the next set of terminologies. The next term is self-cross. A self-cross is a cross between individuals of the same generation. Kamu akan nampak perkataan cross. Cross means dibiak. Okay, kita akan biakkan atau kawinkan dua individu atau mungkin lebih dari dua individu. And when we say self-cross, bukan satu individu dengan diri dia sendiri lah. dia merujuk kepada kita kawinkan individu dari generasi yang sama. For example, in this situation, we have a tall plant and a dwarf plant. Kita kasih kawin dua baka tumbuhan ini, dia hasilkan generasi yang semua tinggi. Generasi yang semua tinggi, this is the first generation, jadi kita tulis F1. Kita ambil daripada generasi yang tumbuhan ini semua tinggi, kita kasih kawin-kawin, kasih biak sesama dia, untuk hasilkan generasi kedua. When we are taking that first generation and breeding them with each other, that is self-cross. Okay, and we are going into the next term. The next term you need to know is test cross. Test cross is a cross between an individual of dominant phenotype but unknown genotype with a recessive homozygous individual. Test cross ini kita gunakan untuk test sebab kita ada satu tumbuhan yang kita tahu dia punya phenotype, tidak tahu genotype dia. Cara kita tahu genotype dia adalah dengan melakukan eksperimen yang digelar sebagai test cross. We're going to go into test cross a little bit later, so I'll explain it more in the next slides. Okay, third term in this slide that you should know about is true breeding or pure breeding. Ataupun kadang-kadang ditulis sebagai true bred, pure bred. True breeding individuals are individuals with homozygous genotype, either dominant or recessive. Kita boleh katakan dia adalah baka yang tulen, okay, keturunan yang tulen. So in this example, we have two pure breeding individuals. This one with big T to produce tall plants is a pure breeding tall plant. This dwarf plant with small t is purebred dwarf plant. Okay, asalkan dia punya genotype adalah yang sama, kita boleh gelar dia sebagai pure breeding, tulen. Okay, we'll talk a little bit more about genetic diagram. So, kalau kamu belum sedia maklum, ini digelar sebagai genetic diagram. This is called a genetic diagram. It helps us to trace from the first generation or the parent generation, bila kita kasih kawin dia, macam mana anak dia. Antara generasi pertama ni, bila kita kasih kawin lagi, apa macam anak dia. Kasih kawin lagi, apa macam anak dia. That is the use of a genetic diagram. Okay, as you see here, we always start with P, which stands for parent generation, ataupun P generation. Kamu akan nampak dalam slide kamu dan dalam banyak nota-nota dan mungkin video-video pencara lain, university lain, dia akan tulis P sahaja. Tapi, saya galakkan kamu, tolong tulis generation. Sebab ada beberapa tahun semasa PSPM, pelajar tidak dapat markah kerana mereka cuma tulis P. Dia tidak tulis parent, dia tidak tulis generation. Dan masa itu, tahun itu, pemeriksa agak tegas, mereka tidak diberi markah. Untuk keselamatan, kami galakkan kamu tulis P generation. Okay, so the first we start with P generation atau parent generation. Di sini parents dia adalah one tall plant with genotype big T, and one dwarf plant, satu tumbuhan kerdil with small t genotype. When we are drawing the genetic diagram, we need to take out the gametes. G stands for gametes. So the gamete from this is big T saja. The gamete from this is small t. The gametes must always be drawn in a circle. And then you will breed them, okay? Kasih gabung, kasih cross dia, untuk hasilkan F1 generation. Or sometimes it is written as F1 progeny. it just means generasi pertama. And you write here big T small t, sebab sini big T small t, and you produce all tall phenotypes. For self-cross, kamu boleh tulis F1 times F1 macam ini, ataupun kamu boleh tulis F1 self-cross, tapi tidak perlu tulis generation. So, begini saja pun okay lah for this one, for this step. When you want to do self-cross, okay, kita gunakan genotype dari atas ni. So, big T small t, cross with big T small t. We take out the gametes. So, as you see, if it's homozygous, satu saja gametnya. If it's heterozygous, it will produce 2 gametes each. Okay, so this one produces big T, small t. This one also produces big T, small t. And when you cross them with each other, you will form the F2 generation ataupun second generation. Okay, so ingat, kalau mampu, sila tulis full untuk mengelakkan kamu kehilangan markah. Something else that you should pay attention to is you should draw or write genetic diagrams row by row. Okay, tulis satu baris demi satu baris supaya kamu tidak ketinggalan apa-apa maklumat. Jangan dapat saja kertas terus tulis T times T, 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 T. Semua tulis sebelah kanan, tapi lupa label P generation, lupa label gamete, lupa label F1 generation. Kalau ingat nanti mungkin kamu boleh bubuh di tepi, tapi nanti hodoh lah, satu ternaik sikit, satu terturun sikit. So, best if you can practice, do it row by row. Satu baris demi satu baris. And please remember that you need to draw or write your gametes in circles. Asalkan kamu kasih keluar gamete, pastikan kamu bulatkan. Okay, kalau tidak bulatkan, dia tidak dianggap sebagai gamete. And where possible, please write the genotype and the phenotype together. Asalkan kamu ada tulis dia punya genotype, cuba untuk sisipkan phenotype dia berdekatan. Okay, di atas kah, di bawah kah, it's not such a big deal, but you should definitely write it together with the genotype. Okay, let us move on to test cross. Okay, ini ada yang saya cakap test cross tadi. Test cross is used to test whether the dominant phenotype is homozygous or heterozygous. Jadi ini adalah contoh. Contoh kita dapat satu pokok. Pokok ini dia punya phenotype adalah dia pokok yang tinggi. It's a tall pea plant. So we know it has a dominant allele, at least one dominant allele, dia kemungkinan T besar, ataupun T besar, T kecil. Macam mana kita mau dapatkan kepastian adalah kita ambil tumbuhan yang tall ini, this tall individual. We are going to cross with a homozygous recessive individual, or in this case, it will be a dwarf plant. Why? Because the dwarf plant will only produce recessive allele. Bila kita kasih keluarkan gamete dia akan nampak macam ni. If it is a dominant, homozygous dominant plant, okay, kalau plant yang kita tidak tahu dia punya genotype tu, if it is homozygous dominant, first generation will be all tall plants. If the plant that we are testing, if it is heterozygous, means dia ada satu T besar, satu T kecil. Bila kita kasih kawin dia akan hasilkan ada dalam generasi pertama tu ada yang tinggi, ada yang renek. Okay, it may produce some tall offspring, some dwarf offspring. Basically, when we do a test cross, Depending on the results of the F1 generation, we can figure out whether the parent is big T or big T small t. Kalau sekarang kamu belum berapa dapat tangkap konsep ini, jangan risau, bila kita buat latihan, kamu boleh dapat gambaran yang lebih jelas dan harap-harap dapat terus ingat dan terus paham. Okay, so those are the terms that we needed to learn. Don't worry if they feel like they're a lot. By the time that you finish this chapter, those terms feel like very kacang to you. Speaking of kacang, let's go into Mendel's experiment. So Gregor Mendel is, you can consider him as the father of genetics. Okay, dalam bidang genetik ni dia antara saintis pertama yang keluarkan teori dan cuba memahami genetik. Okay, so who is this guy? He was born in 1822. He passed away sometime in 1884, and he was a monk who bred pea plants. Monk means Sami, tapi ini bukan Sami Buddha lah, ini Sami Christian. Okay, and the thing about monks is Sami-Sami ni biasa dia ada kawasan dia tu, ada tempat mereka tinggal dengan satu sama lain. Besides praying whatever they want to pray, they also plant gardens, they clean the place, they cook, they write books. So for this person, this Gregor Mendel, he was in charge of the garden. And dalam dorang punya taman tu adalah kacang pea. Dan dia yang kasih biak kacang pea itu. He has a scientific mind. Okay, so although he is a religious person, he also has a very scientific approach. He effectively applied quantitative methods to the study of inheritance. Dia sendiri tengok, ya kan? Kadang-kadang saya kasih kawin begini, dia boleh hasilkan hasil yang begini. So dia dia buat eksperimen sendiri. Kaedah dia jalankan eksperimen tu, dia pastikan dia menggunakan kaedah yang kuantitatif, yang boleh dinilai. And basically, he collected all his data, and we learn a lot more about inheritance. If you want, you can go and find stories about Gregor Mendel. I'm sure they have a lot of stories about the father of genetics on the internet. And I'm sure they have nicer diagrams, nicer pictures than me too. So feel free to go and learn more in your own time. But yeah, we are going to focus more on the peas that he used. And why did he use peas for his experiment? Kenapa kacang peas? Ada 4 sebab utama kenapa dia gunakan kacang pea untuk dia punya eksperimen. Number 1 is they're very easy to grow. Okay, senang untuk tanam. They have short life cycle. Jadi dia tidak payah tunggu 10 tahun baru dia nampak generasi pertama macam mana, generasi kedua macam mana. And number 3, their pollination could be controlled. It's important to have plants with easy to control pollination. So you know exactly what you cross them with. And number 4, they have easily observable characteristics. We're going to look at the characteristics of the pea plants that Mendel chose. Okay, tadi point keempat, they have easily observable characteristics. These are the characteristics of the pea plants. Inilah ciri-ciri yang ada pada kacang-kacang peas yang Mendel pilih. Ciri-ciri pertama adalah seed shape. The first characteristic is the seed shape. The seed can be round or it can be wrinkled. Dia bulat atau dia berkedut. The seed can be yellow, they can be green. The flower on the plant can be purple, can be white. The pod, okay, I don't know what you translate pod, but the thing, and then you open inside the thing, baru ada itu, kacang. Okay, the lapisan salutan luar tu, that is the pod. Okay, the pod can be inflated or smooth, and sometimes they are constricted. So, kadang dia bulat macam ni cantik, kadang dia berkedut. The pod can be yellow, the pod can be green. And Mendel also looked at the flower position. Kadang-kadang itu bunga ada di tepi, axial, at the axis. Kadang-kadang bunga dia ada di hujung saja, di terminal. And last but not least, he also looked at the stem length, or sometimes referred to as the height of the plant. Okay, ini kadang-kadang kita ada kacang pis yang membesar dia bertumbuh, dia boleh tinggi. Tapi kadang-kadang kita dapat kacang pis yang redneck ataupun dwarf. Okay, so these are the 7 characteristics of pea plants that Mendel observed. And as you can see, it's pretty easy to distinguish between the dominant trait and the recessive trait. Tolong ingat yang ini ya. Tolong ingat apa 7 ciri-ciri dia. Dan mana satu dominant, mana satu recessive. Penting nih. Okay, so yeah, let's just move on from the pea plants. We'll talk a little bit more about how did Mendel do his experiment. Memang Mendel ni dia akan ambil satu parent, dua parent, and then kasih biak. In a typical breeding experiment, Mendel will start by crossing 2 true breeding individuals as the pea generation. And there will be 2 types of crosses. There will be monohybrid cross and dihybrid cross. Okay, dia ada 2 jenis eksperimen. Satu adalah pembiakan monohybrid, kedua adalah pembiakan dihybrid. Monohybrid means he is crossing or mating or breeding the plants, and he only involves one character determined by one gene. Bila dia buat monohybrid cross, dia cuma tengok satu ciri-ciri. So, contoh dia kita tengok ciri-ciri stem length. For monohybrid cross, we only look at whether the plant has tall stem or dwarf stem. In a dihybrid cross pula, it will be the crossing that involves 2 characters determined by 2 different genes at different locus. For example, it involves color of flower and stem length. So kita tengok 2 ciri-ciri pada masa yang sama. Okay, so let us go into the very first type, which is the monohybrid cross. We'll first talk about a monohybrid self-cross. Okay, we're going to start with, macam saya cakap tadi, biasanya Mendel dia akan mula dengan 2 purebred. One is dominant and one is recessive. So this one, he is taking tall purebred plant and dwarf purebred pea plant. We start with the parent generation, tall plant cross with dwarf plant, and then we take out the gamete. Tulis gamete, T huruf besar, T huruf kecil, buat dalam bulatan. And then we cross, kawinkan dia. Dia akan hasilkan first generation, big T, small t. In this first generation, or F1, all the plants in F1 are tall because it is heterozygous. Then we are going to do a self-cross. In the self-cross, we take big T, big T, small t, cross big T, small t. Kita keluarkan gamete dia. Gamete dia, big T and small t in different circles. Jangan lupa lukis anak panah. And then we do the crosses for F2. Okay, I'm sure you've done this in bio in school before, but in case you forgot, this is how you do it. You draw this line to this line, okay, and then you get homozygous dominant tall plants, okay, and this one with this one, you get heterozygous tall offspring. With this, you get another heterozygous tall offspring. This gamete with this gamete, you get a homozygous recessive offspring. And yeah, so these are the results of your F1 crossless. When F1 plants are self-crossed, the F2 or second generation showed that 787 plants are tall, 277 are dwarf. So, yang ditulis dalam bahagian yang hijau ini adalah data daripada eksperimen Mendel sendiri. Okay, bila dia buat eksperimen tu, of course dia kasih tanam sampai beribu pohon tumbuhan yang dia tanam, dan dia kira satu-satu. Dia boleh nampak 787 adalah tall, 277 dwarf. Kamu boleh dapatkan nisbah daripada ini. Ratio means nisbah, so you can get a phenotypic ratio of 3:1. So the genotypic ratio you can get from the genetic diagram. The phenotypic ratio, you look at the phenotype. Kalau ada yang kurang jelas macam mana mau dapatkan nisbah, boleh tanya, boleh rujuk nota-nota maths juga. Okay, so let's look at how does it look for a monohybrid test cross. The genotype for this tall pea plant may be homozygous dominant or heterozygous. So a test cross is used to determine the unknown genotype. Okay, so this one we are showing from tall plant. If it is a tall plant crossed with a dwarf plant, tall homozygous. If the test cross is done on an individual which is heterozygous for one character, the phenotypic ratio for the offspring will be 1:1. Okay, contoh daripada ini, kalaulah kita mau tentukan sama ada yang tall offspring ini adalah homozygous dominant ataupun heterozygous, kita cross dia dengan yang homozygous recessive. Tengok dia punya offspring, anak dia dapat macam mana. Kalau anak dia menunjukkan phenotypic ratio 1:1, okay, 1:1, that means the genotypic ratio will also be 1:1, and that means that the parent was heterozygous. Kalau dia homozygous, anak dia semua akan tunjukkan phenotype yang sama. Okay. Alright, so we are also going to go into Mendel's laws of heredity, and which is in Mendel's first law of segregation. So what is Mendel's laws of heredity? Ini adalah hukum-hukum Mendel. We're going to go into the first law, the law of segregation. Mendel's law of segregation states that an organism's characteristics are determined by a pair of factors or alleles. Of a pair of such factors, they can only be carried in a single gamete. Ini menyatakan ciri-ciri sesuatu organisme itu ditentukan oleh sepasang faktor. Dulu, zaman Mendel, bila dia buat eksperimen ni, diorang belum tahu lagi apa itu gene, diorang tidak tahu apa itu allele. Itulah sebab kenapa Mendel menggunakan perkataan factor. He didn't have the word gene, he didn't have the word alleles, which is why he used the word factors. But with what we know now, we know that he was actually describing alleles. So, apa yang dia katakan adalah satu organisme ada dua allele. Bila dia hasilkan gamete, allele itu akan terpisah. Satu gamete cuma akan bawa satu allele untuk satu gene. Okay, so he postulates that the height of plant was determined by factors. Okay, the height of his pea plants was determined by alleles. These alleles or factors will come in pairs, memang berpasang. The F2 generation contain only tall and dwarf, no intermediate. Okay, apa yang Mendel nampak adalah phenotype dia antara tinggi atau pendek. Tiada pertengahan, no intermediate. So the F1 generation must contain both tall and short factors, even though all the plants were tall. And the first factor is a dominant allele. So apa yang Mendel tulis ini adalah allele yang ditulis dahulu adalah yang dominant. Contoh P besar, P kecil. P besar adalah ditulis dahulu dan itu adalah dominant. The second factor yang ditulis di belakang akan menjadi recessive allele. Okay. During Mendel's time, he didn't really know about meiosis, but now we do. So we try to relate the law of segregation with what happens in meiosis. So we know that the segregation of allele will occur during meiosis 1. That is when the homologous chromosomes will pair up and then exchange genetic material. They will randomly align on metaphase plate and then separate. Okay, kenapa satu gamete cuma membawa satu allele? Disebabkan semasa meiosis 1, setiap allele itu akan terpisah daripada kawan dia. Itu sajalah. Okay, so when we talk about monohybrid, you should be able to draw monohybrid genetic diagram. You should be able to do monohybrid test cross. And you should be able to describe what is the law of segregation and what is the relationship between law of segregation and meiosis. Okay, nanti kita buat latihan, kamu akan lebih paham. Okay, so that is it for monohybrids. We're going to move on to the second type of crosses, which is dihybrid cross. Okay, so we're going to start with a dihybrid self-cross. So in one experiment, Mendel will cross plants from 2 true breeding strains, one with tall purple flowers, the other dwarf with white flowers. So this is dominant phenotype, recessive phenotype, recessive phenotype. All the offspring in the first generation were tall with purple flowers. These will be the dominant characteristics. Okay, let's see what I just wrote in genetic diagram form. Kita ambil maklumat yang ditulis tadi, kita tengok dia dalam bentuk genetic diagram. So, ayat tadi adalah Mendel akan ambil tall purple flower cross with dwarf purple flower. Itu adalah P generation. Kita keluarkan gamete. Bila kita keluarkan gamete untuk dihybrid, pastikan dalam satu bulatan itu ada 2 gene punya allele. Sini ada T, ada P. Jadi gamete kamu pun patut ada T, ada P. Jangan dua-dua T, mesti berbeza. On this side, small t, small p. The gamete produced is small t, small p. Kasih kawin dan hasilkan first generation. So first generation dia akan jadi gabungan. Big T, small t, big P, small p. Huruf yang sama dikumpulkan bersama. Yang dominant mesti di depan. Yang resesif mesti di belakang. So you can see here big T, small t, big P, small p. Jangan tiba-tiba T besar, P besar, T kecil, P kecil. Itu adalah salah. Okay, so pay attention to your notes. Okay, after we get the first generation will be self-crossed and produce the following phenotypic ratio in the F2 generation. Generasi pertama self-cross, kawin, kira-kira. F2 generation akan hasilkan nisbah ini. They will produce this ratio, which is 9 tall purple flower, 2, 3 tall white flower, 2, 3 dwarf purple flower, 2, 1 dwarf white flower. 9, nisbah 3, nisbah 3, nisbah 1. Okay, let us see that in diagram. Okay, kalau kita buat self-cross di sini, dia akan hasilkan 4 jenis gamete. So, 4 jenis gamete tuh T besar, P besar, T besar, P kecil, T kecil, P besar, T kecil, P kecil. Same thing on the right side. Now, if it's monohybrid, kalau monohybrid senang lah kan kita lukis garis-garis. Tapi dihybrid macam banyak sangat garis. Jadi kita akan buat Punnett square. Punnett square nampak macam ini. So you take all the gametes from this parent. Okay, susun di sebelah sini. All the gametes from this individual susun sebelah sini. Dan pandai-pandai lah kamu kasih match. Dan perlahan-perlahan isi jadual Punnett square. Okay, so after you fill it in, then you see one by one what is the phenotype. Ini adalah genotype dia. Kamu tengoklah satu-satu. Kalau dia T besar, P besar, P besar, dia adalah tall purple. Kalau T besar, T kecil, P besar, P kecil, masih juga tall purple. T kecil, P besar, dwarf purple. Satu-satu kamu tengok. Okay. Sudah tengok dia punya phenotype, kira, simplify, kasih jadi nisbah, dan kamu akan dapat phenotypic ratio 9 to 3 to 3 to 1. When Mendel was doing this experiment, he repeated the dihybrid cross experiment for other pairs of characters and always observed a 9 to 3 to 3 to 1 phenotypic ratio in the F2 generation. Maksud ayat ini adalah dia buat berulang kali eksperimen ni, dia dapat juga nisbah 9, nisbah 3, nisbah 3, nisbah 1. Dan setiap ciri-ciri ini diwarisi independent. Okay? Kalau dia tall plant dengan dia punya purple flower, dua ciri itu tidak mempengaruhi antara satu sama lain. That is what is meant by inherited independently. Okay. We're going to go into more dihybrid. We're going into dihybrid test cross. So, a genotype for a plant with tall purple flower may be big T, big P, which is homozygous dominant for both traits, or big T, small t, big P, small p, or what we can say, heterozygous for both traits. How we know if the tall plant with purple flower is this genotype or this genotype? We do a test cross. If the test cross is done on an individual which is heterozygous, the phenotypic ratio for the offspring will be 1 to 1 to 1 to 1. Okay, let us see that in an experiment. Kalau kita ambil yang memang T besar, P besar, kita buat test cross. Test. Okay. Anak dia akan jadi semua phenotype yang sama, yaitu semua tinggi, semua purple. Tapi, kalau kita buat test cross untuk individu yang heterozygous, if we test cross a heterozygous individual, it will look like this. This one you can do a cross because it is still simple. You can also do a Punnett square. These are the genotypes you will get. And these are the phenotypes. You will get 1 ratio tall purple flower, 1 ratio tall white flower, 1 ratio dwarf purple flower, and 1 ratio dwarf white flowered. Okay, so if you can see, if it's heterozygous, it will get 1 to 1 to 1 to 1 ratio. Okay, so based on that, you will know whether the original plant is homozygous or heterozygous. Okay, so when we talk about dihybrid cross, we also talk about Mendel's second law of heredity, which is a law of independent assortment. So Mendel's second law of independent assortment. It states that each member of a pair of alleles may combine randomly with either one of another pair of alleles during gametogenesis. Dalam arti kata lain, satu allele tidak akan mempengaruhi posisi atau proses pewarisan allele yang lain. And we also talk about the relationship between law of independent assortment and meiosis. zaman Mendel dia tidak tahulah macam mana proses meiosis itu, tapi sekarang kita gabungkan teori Mendel dengan apa yang kita tahu. Okay, what we know is by the end of meiosis, the genes on pairs of homologous chromosomes have been sorted out for distribution into one gamete to another independently of gene pairs of other chromosome. Ini lebih kurang apa yang saya cakap tadi. Gene pada kromosom yang berbeza tidak akan mempengaruhi posisi gene pada kromosom lain. Diorang sendiri-sendiri, tidak kacau, tidak ganggu, tidak mempengaruhi. Okay, Mendel, he states that assortment of one pair of alleles is independent of that of another pair of alleles. Ayat yang lebih kurang sama. This is only true if the two pairs of alleles are located at different chromosomes, not linked gene. Okay, so hukum Mendel yang kedua ini, law of independent assortment, dia benar kalau gene yang kita tengok itu terletak pada dua kromosom yang berbeza. Kalau dua gene yang kita tengok itu terletak pada kromosom yang sama, dia tidak lagi ikut hukum Mendel. Benda-benda yang tidak ikut hukum Mendel, kita akan belajar dalam 4.2. So we're going to learn about the phenomena that doesn't follow Mendel's law of inheritance in 4.2, which is next week's material. But for now, we are going to finish with law of independent assortment and meiosis. Okay, so just a quick summary. We already finished this subtopic, which is 4.1 Mendelian genetics, monohybrid and dihybrid. We talk a lot about the terminologies, and we talk about monohybrid crosses and dihybrid crosses. Of course, listening to me talk is not going to help you understand everything about this chapter. So what should you do now? You should definitely do some exercise. Okay? Chapter ni memang kita perlu buat latihan dan kamu perlu semak balik diagram kamu untuk kamu betul-betul faham apa yang kamu belajar dalam chapter ni. So please, now, complete the 4.1 monohybrid and 4.1 dihybrid worksheets, which you can find in your Google Classroom. We will discuss these exercises soon within this week. So that is all from me. Thank you very much for your time and attention, and I will see you again in the next lesson. Thank you. Bye.